Hello! In this video, I will demonstrate how to analyze your single-cell multiome data using SciDAP. Here I created a new project, and now we can add the pipelines to it. We click Edit Project, and here you can see all the pipelines available. I already selected a few that we will need for the analysis. Now let's return to our folder and add the first sample. Single-cell multiome analysis is based on a tool called CellRanger, and the first step is creating the reference genome index. Here you select CellRanger reference pipeline, which will create the indexes for you, and give it a name. In my case, I will be using the human genome version 38, and simply select Save and Run the sample. In the next video, we will review how to upload your input files.